Hello student, our next topic is prokaryotic cell if we talk about the IB curriculum biology. So prokaryotic cell, it is a very simple I can say topic. If we talk about the structure of the prokaryotic cells, you must be known that these cells divide into two types according to the structure. If we simply talking about any cell, it can be either prokaryotic or it can be eukaryotic. The first cell to evolve were prokaryotic, and many organisms still have prokaryotic cells, including all bacteria. Okay, when the life originated on earth, we assume that the first organisms which came into life were prokaryotes. Okay, in simpler language, if we talk, all the bacteria belong to this category of prokaryotic organisms or prokaryotic cells. They have a very relatively simple structure as compared to the eukaryotic cells. As you can see, and you will further see also, that eukaryotic cells, they are divided by the membranes into separate compartments such as nucleus and mitochondria. But here in this case also you can see the drawing of the prokaryotic cell. It is a simple structure containing cell wall, cytoplasm, nucleoid, which is a DNA containing region in case of prokaryotes. External pili or hair-like structures are present that helps in the movement and attachment in some of the cases, and the ribosome that is present, that is of 70S type. There is one flagellum also in this diagram, you can see. So flagella is also going to help in the mobility, whereas this pili structure, that is going to help in reproductive part also, that you will further study in your further units. Like prokaryotic cells, they are not compartmentalized. Okay, in eukaryotic cell you can see so many compartments present in a single cell, like ribosomes, Golgi apparatus, lysosomes, nucleus. But in case of the prokaryotic cells, only simpler structures like cell wall, cytoplasm and nucleoid is present. So nucleoid is the structure in prokaryotic cell where DNA material is present, and no clear cut membrane is present that can divide the distinction between the cytoplasm and the nucleus. Okay, here are few pictures also. Salmonella bacteria showing their internal structure under the microscopes. Now binary fission in prokaryotes, if you talk about the division or the cell division in prokaryotes, that is going to take place mainly by the binary fission process. Okay, binary fission, it is the simple division of the cell into two equal parts. We can say a single organism is going to divide into two daughter nuclei. This simply means splitting into two. So the bacterial chromosome is replicated, so there are two identical copies. So first of all the chromosomal material or DNA material is going to double in size or make the identical copies. These are moved to the opposite ends of the cell and the cell wall, plasma membrane, and they are pulled inward, so the cell pinches apart to form two identical cells. Here you can see. So in the middle center, that you can see the pinching, that is the start of the division. Okay, first they have divided the material, then in the center there is some pinching kind of structure that is going to divide into its daughter nuclei or the identical cells. So some prokaryotes can double in volume and divide by binary fission every 30 minutes. Okay, so cell division in lower organisms or prokaryotes, it's very fast. Escherichia coli, like here it has given the example, it takes only 20 minutes to divide. In 20 minutes all the DNA material can be doubled, all the sharing you can say cell organelles, they are going to be divided just only in 20 minutes. Okay, and mostly bacteria, they take the time of 20 to 40 minutes to perform their binary fission. So that's all about the prokaryotic cell. I hope you have understood these topics. So this is an overview of the prokaryotic cell, their structure and their division part, that is the binary fission. Thank you, students.